a lot of people don't realize is that high energy electrons run all living things.
You've just described a three-part pathway. You've got NMN, NAD, ATP.
NMN doesn't go into cells.
Didn't they just recall this a few months ago?
All fresh food has NAD precursors because, like I said at the very, very beginning, NAD is the electron carrier in all of life.
Charles.
Louisa.
How are you?
I'm very well, thanks.
I've been following you for a long time, so I'm super excited about this. Your space intrigues me, albeit, in the neurology space. I'm now going deep into the longevity space and been following along your tweets and your wonderful threads, and I'm really intrigued by them.
Good.
And I'm sure the audience is too.
Great.
So why don't I first want to understand how you even got into the field of metabolism and understand what intrigued you about this area.
Well, I guess I've been working as a professional, molecular biologist for 40 years. That's the short answer. I can give you the whole path if you want.
I'm excited about the whole path.
Let's see. So I'm 61. I'm going to be 62 in a couple months. Graduated from college in 1983 with a degree in biology. Moved to California from Connecticut, and worked in biotech for 5 years in a couple of different companies. And then I moved to Stanford, got my PhD in Stanford in the cancer biology program, biochemistry department. I was working on pro-hormone processing. So back in the day, in the late 80s, early 90s, when I was working on it, nobody knew what was the enzyme in our cells that's responsible for processing insulin, right? You know that insulin is made from a precursor molecule, right? It turns into an A chain and a B chain. The C peptide is released, right? All that stuff. So there's a cutting enzyme called a protease, and we actually purified that protease. We purified the yeast form of that protease, but it's essentially the same enzyme as humans have. So that's what I got my PhD for. 1993, had my PhD. Spent 10 years in Northern California. Decided to go back to Boston. In my field, you're not considered a fully formed scientist with a PhD, believe it or not. You know this. So did postdoctoral training at Brandeis, and there I added X-ray crystallography to the things that I do. I was funded by the Leukemia Society at Brandeis for a 3-year postdoc. Started working on a family of enzymes that have to do with nucleotides.
Yes.
Like the building blocks of DNA and RNA, building blocks of NAD. NAD is a nucleotide itself. And so I had a successful postdoc, got my first faculty position in 1996, really focusing on these nucleotide binding proteins and nucleotide-related enzymes. And in my first 6 or 7 years as an independent faculty member, I ran into NAD. I ran into NAD, the central catalyst of metabolism. And 96 to 2003, I was in Philly at Thomas Jefferson University. 2003, I moved to Dartmouth College, Dartmouth Medical School. And that's where I really kind of blew open the NAD thing.
Seemed more exciting than the cancer biology?
Well, to me, my current title at City of Hope, I skipped a little bit there. There's some exciting stuff in the last 20 years since 2003 when I moved to Dartmouth. But right now, I'm a chair of a department called Diabetes and Cancer Metabolism. So NAD relates to cancer. NAD relates to diabetes and obesity. It relates to rare diseases. It relates to mitochondrial diseases. It relates to health. It relates to neurodegeneration. It relates to heart failure. NAD is, we should say what NAD is,
well,
right?
I was going to, because metabolism and metabolic health being the central theme of this entire episode and being the central theme of neurodegeneration and longevity, why don't we tap into that?
Okay, we'll do the last 20 years at some point.
Yeah.
But so, all right, I drove here in an electric vehicle, and you'll see where I'm going in a minute here. But most people have an idea of what's inside an electric vehicle, which is a huge battery, right? And then a lot of moving parts. So you have transmission, you have a steering wheel, you have a cooling system, you have an entertainment system, you have a braking system, right? All of those things require electrical power. They require power, and the power is transmitted on copper wires, right? And you push the button and you have current, right? And you could deliver, high energy electrons to all of those different components.
Mm-hmm.
So what a lot of people don't realize is that high energy electrons run all living things, not just electric cars, but high energy electrons run plants and animals and fungi and bacteria and everything else. And the energy from high energy, the energy in the high energy electrons actually comes from the sun. We could go into that if you want, but it basically is captured onto something called NAD. So in metabolism, we have protein, fat, and carbohydrate, which are the three macronutrients from which we derive our calories, right? Which is another kind of energy conversion unit. And in the course of burning that fuel, we capture high energy electrons onto a molecule called NAD. And when the electrons are captured, the form of NAD is called NADH, right? At the inner mitochondrial membrane, the NADH electrons initiate the electron transfer chain. That generates ATP. That generates most of the ATP in our bodies. In our skeletal muscle and our heart, you generate phosphocreatine, which allows us to have the actin, myosin, gymnastics and, power our muscles and power, blood circulation. But ATP is required for, virtually all of the energy-dependent steps in metabolism. It's required for us to transmit, ideas. It's required for us to record memories. It's required for us to have emotions. It's required for us to do repair reactions. So that's how central NAD is. NAD, NADH is centrally involved in cellular bioenergetics, converting our food into energy, right? And then there's actually two other NAD coenzymes that a lot of people don't even mention, but they're called NADP and NADPH, right? And they're required for anabolic reactions. So they're required...
without oxygen?
No, anabolic means making stuff, like building stuff. We use the term anabolic steroids. That means, a hormonal system that's involved in making things. So we have to make our own lipids, right? We make the bulk lipids for, cell membranes, right? If you repair cells, you have to make cell membrane. You're turning over RNA all the time. You have to replicate DNA, so you have to make DNA, RNA, protein, and lipids, right? All of those reactions require electrons on NADPH.
Okay.
And there's also detoxifying reactive oxygen species. Depends on electrons on NADPH. So what our group has discovered, really, we're known for two things.
Mm-hmm.
So shortly after the move to Dartmouth, when I realized that there are a lot of unsolved problems in NAD synthesis, even though NAD was a classically known nucleotide and classically known as the carrier of high energy electrons, I started realizing around 2003 to 2004 that there was a very good possibility that there was another way of making NAD that hadn't been investigated yet. So I discovered the nicotinamide riboside kinase pathway. So discovered the vitamin activity of NR, and my postdoc and I, Pavel Biagonovsky and I, discovered the identity of the nicotinamide riboside kinase genes and the nicotinamide riboside kinase enzymes that are used to convert NR into NMN and subsequently NAD coenzyme. So we're known for this NR pathway.
NR being the precursor.
NR being, it's actually the biggest piece of NAD that can get into a cell and replenish NAD coenzymes, right? Now, the other thing that our group is known for is we developed a technology called quantitative NAD metabolomics. It's a lot of syllables, I'm sorry. But you know about genomics, right?
Mm-hmm.
So with genomics, we can sequence anything that we want, right? Chris Mason, maybe someday, he's in New York, you can interview Chris, and he goes around like swabbing subways and going to various places and sequencing all kinds of organisms, right? Sequence bacteria off of, you can imagine subway seats, and so you can sequence anything, right? Yeah. You can sequence us, you can sequence, tumors, you can sequence tumor evolution, you can sequence novel coronaviruses, you can sequence weird plants and animals that you find in unusual places, you can sequence, microorganisms that you find in deep sea vents. So that's genomics, right? And there's transcriptomics, right, which is characterizing RNA species, right? And there's proteomics, which is characterizing proteins, all the different forms of proteins. And then there's metabolomics. And metabolomics is really, the genome is relatively stable, right? In a cancer cell, it's not as stable. In certain immune cells, it's not as stable. The antibody genes rearrange and so forth and so on. But in metabolism is always changing, right? So, for example, in your hepatocytes, right, after breakfast, if you're a breakfast person, your hepatocytes are oxidizing fuel, right? But overnight, when your hepatocytes are responsible for keeping your blood glucose up, they are taking things like malate and oxaloacetate and making glucose, right? So they're running reactions in different directions. They're running different kinds of reactions, right? It's almost like there's a famous chapter, called Ecclesiastes, and the Byrds made a song out of it back in the day. What is it? There's a time to sow and a time to mend, a time to burn, time to build up and a time to tear down, right? That's the way metabolism is, right? Metabolism has times when it's oxidizing fuel, that's catabolic. Metabolism has times where it's making stuff, that's anabolic, right? And so when it's catabolic, it's taking energy that we eat or that we've stored, right? And it's harvesting these high energy electrons onto NAD coenzymes. And when metabolism is anabolic, making stuff, we're actually taking those high energy electrons and using them to do stuff like make androgens, make estrogen, make cholesterol, make cell membrane.
Wow. There's a lot to unpack there, because what we usually hear when it comes to cell energy metabolism is ATP.
Yep.
We rarely hear, obviously if you want to be extremely nuanced and talk to a professor like yourself, you've just described a 3-part pathway. You've got NMN, NAD, ATP.
NMN, so I knew we'd get to NMN at some point. So there's really 4 ways. So eukaryotic organisms are what we're talking about. Animals are eukaryotes, right? Mammals are eukaryotes. And bacteria are kind of a separate thing, right? But there are genes and enzymes to convert 4 different things into NAD and the 4 NAD coenzymes: NAD, NADH, NADP, NADPH. So there's something called the de novo pathway that starts with tryptophan. Okay, de novo basically means you're making it from scratch, right? It's like if you were making a cake, or you're making I'm going to make pizza tonight. So I'm doing a de novo synthesis of pizza.
That's the same as because I've read a lot about de novo lipogenesis.
De novo lipogenesis, right. So de novo lipogenesis means that you're starting out with starting materials that look nothing like fats.
Yes.
Like literally, it could be sugar, right? You can take sugar, run glycolysis, and of course we're very good at this because we're very good at taking something like sugar, and there's only so much that we can store as glycogen. Well, glycogen is like polymerized sugar. Yeah. So that's not really a de novo pathway. That's picking up a unit and kind of polymerizing it, right? Mm-hmm. That's more of what's called a salvage synthesis. Lipogenesis, you're taking sugar and you're breaking it down, right? And it's broken all the way down to something called pyruvate, and then it goes into mitochondria, and then there's citrate formation. And citrate, and I don't know if the details are important to people or not, but the citrate in the mitochondria goes into the cytosol and becomes cytosolic acetyl-CoA, and then that goes to cytosolic acetyl-CoA and cytosolic malonyl-CoA, and then you can make fat, right? So there's a way to take sugar and make fat, right? And it's a de novo pathway because the intermediates look nothing like the starting materials and the product looks nothing like the starting materials. So that's how our liver and a few other tissues can make NAD from tryptophan. If we got out a whiteboard and I drew the structure of tryptophan and then I drew the structure of NAD, you would be hard-pressed to find more than 2 atoms in a row in tryptophan that you find in NAD. So that's a de novo pathway, takes 8 enzymatic steps. Okay. It actually takes a lot of ATP as well to convert tryptophan into NAD. And there's many other things that tryptophan is a precursor to. Like tryptophan is used to make protein.
Exactly, yeah.
Tryptophan is used to make serotonin, right? And tryptophan goes through all these so-called kynurenine metabolites, and some of them go to NAD and some of them go to other things, okay?
Yeah.
But the other way is what's called salvage. And salvage, so just to go back to my pizza tonight, I started last night with flour, yeast, water, salt. Okay, the classical, Napolitano thing, okay? I guess if I really was going to Novo, I would have some ripe tomatoes as well. But I do have, a jar of, or a can of tomatoes that are going in. But totally de novo means totally from scratch. Okay, salvage means that you stop off at Trader Joe's and you get a package of dough, or you get a rolled out dough. You get a salvageable unit. You get a usable unit of component, a sizable component of that pizza, right? It's a difference between making a car from small units versus going to a chop shop. If everybody knows what a chop shop is, that's where there's like a market for like stolen cars or something like that, or wrecked cars, and a chop shop will like pull the things out that can be sold again, right? So salvage would be using those components, right? And biology is very conservative in the sense that if there's something of value that can be used again, it's going to be used again, right? So there's something called so NAD is a dinucleotide. ATP is a mononucleotide. It's just one nucleotide. Mm. It has one base, it has one sugar, it has phosphates. Mm. NAD has two different bases. It has an adenine base, which is the same as an ATP, the base in an ATP. And then it has what's called a nicotinamide base. And then there's a ribose on both sides, and there's a phosphate on both sides. So like left to right, depending on left to right, right to left. There's nicotinamide, ribose, phosphate, ribose, adenine. That's it. Nicotinamide, adenine, dinucleotide, right? So the most valuable and unique piece of NAD that can be salvaged is called this nicotinamide base. And we call it a vitamin. Mm. So it was discovered in 1938 by somebody named Conrad Lvhem. It's kind of a gory story of how he discovered it. But you can buy nicotinamide in the store, and your body loves it, your cells love it, and it goes into cells, and there's an enzyme. It's called NAMPT, and it takes nicotinamide and turns it into NMN inside the cell. NMN is actually nicotinamide riboside with a phosphate on it. Mm-hmm. NMN doesn't go into cells.
Right.
So it doesn't go into cells because it has a phosphate on it. Right. So, but the history of science is weird. And, about...
If it doesn't go into the cell, then what happens?
So basically, like, you could buy NMN. Yeah. It's not fully legal as a supplement in the United States.
Didn't they just recall this a few months ago?
Yeah, there's an FDA action on it, but you can eat it, right? If you find a safe source of it. What is going to happen is that it's going to be degraded into NR or nicotinamide, and then it's going to go into cells. And then whether it's degraded to NR or nicotinamide, it's going to have to become NMN inside of cells, and then NAD.
So regardless, you're still getting it, somewhat of it, into the cells, whether degraded or not, which then turns into NAD.
And I've used it in the lab. So like our expectation was that, and pretty much anybody's expectation would be that NMN and NR would be equivalent when you do like a rat experiment or a cellular experiment, because the NMN has to be converted to NR in order to act. The prediction that other people made that is false, the prediction was that NMN will just get into cells and then be one step closer to NAD than NR is, right? Because it already has the phosphate. But Carlos Canto, a number of years ago, knocked out the nicotinamide riboside kinase, right? So we knocked out the gene that converts NR into NMN,
Okay.
right? So if you're an NMN proponent and you're saying NMN gets into cells, it shouldn't matter. It shouldn't matter if nicotinamide riboside kinase is knocked out because you're delivering NMN. It shouldn't need nicotinamide riboside kinase.
Correct.
It should go straight to NAD.
Yeah.
But it didn't.
So that was the predicted hypothesis.
That was the prediction of the people that said NMN goes straight into cells.
How could they predict that without using a knockout gene? Like, how could you just predict it without?
Well, you can predict. It's a hypothesis. It's
Okay.
it would be unprecedented.
Yes.
Because it has a phosphate.
Mm-hmm.
So basically, using NAD or NADH or NMN as a vitamin is sort of like using coenzyme A as a vitamin. We use pantothenate, right? So pantothenate is the salvageable piece of coenzyme A, doesn't have a phosphate on it, right? You start pantothenate is great stuff. It's either vitamin B5 or 6, I can't remember. But pantothenate goes into cells, right? And then enzymes convert it into coenzyme A, right? It's great stuff. But people don't supplement with coenzyme A because basically it has to be degraded back to pantothenate, and then your cells have to take the pantothenate up and make coenzyme A. So to me, using NMN as a supplement or intravenous NAD or something like that doesn't really make sense because it has to be degraded to NR anyway. But people are doing it. But basically the biochemistry of it says that NAD and NMN get the phosphates removed, and the biggest piece of NAD that can go into cells is NR.
So explain to me and the audience why this is such a big emerging field. Why is there so much hype around it? I'm now seeing people doing NAD IVs.
Yeah. So do
we want more NAD?
So there's the real reason and then there's the hype reason, and they're not exactly the same.
Okay, I would like the real reason, because there is so much hype, which we're going to get into at the end. But I basically want to know, and I'm sure everyone wants to know, why do we want more NAD, and why does it become somewhat dysfunctional as we age?
Right. So I think I was starting to say, but I got it from this tangent, right? Are tangents okay here?
We love tangents.
We like tangents?
Yeah.
Okay, good. We're doing long form podcasting here. So, but not all day. We're not going 8 hours here. No,
we can,
no way.
Though.
No, but I don't think that we will. So, I think I said, we got really interested in metabolomics, right? Because metabolism is what your cells are doing. So we wanted a way to report omically, like meaning comprehensively, like not sequence one gene, that's gene sequencing, but sequencing all the genes is genomics, right? So we wanted to be able to characterize metabolism omically, and we wanted to do it quantitatively. Okay, so we developed a technology called quantitative targeted NAD metabolomics. Mm-hmm. What came out of that, I think, is fairly astonishing, which is that what I sometimes term as the crown jewels of metabolism, NAD, NADH, NADP, NADPH, are not protected like any crown jewels that you've ever heard of, right? Like, where are the crown jewels kept in any European capital? They're kept inside a safe, inside a vault, protected by armed guards, surrounded by in a castle, surrounded by a moat or something, right? So the crown jewels are highly, highly protected. they're not going to be exposed to the elements. Here we are sitting in the middle of August 2023. We're actually about to get a very unusual storm. I don't know if you're going to be around on Sunday or Monday, but we're expecting a hurricane landfall here in Southern California.
I read that.
The first time in 84 years. And but in metabolism, as we walk around, life, we're exposed to the elements, right? We're exposed to heat. We're exposed to cold. We're exposed to, you just flew in from New York. You have time zone disruption, right? We are living in oxygen, which we need oxygen in order to run the electron transport chain. But oxygen also gives us reactive oxygen species, right? Which have to be detoxified. We love sunlight, but sunlight will damage our DNA, right? We love food, but overnutrition will challenge our NAD system. A lot of people love alcohol and drugs. Those things challenge the NAD system in the liver, in the brain, and other places. Many different disease processes attack the NAD system. So essentially what our technology allowed us to discover is that many different conditions of metabolic stress disturb the NAD system. So for example, when we have sun damage to our DNA, something called pyrimidine dimers form. So 2 T's on the same strand of DNA. It's not uncommon. Like, there's a T every 4th-ish nucleotide, and 1 out of 4 times it's followed by a T. So roughly 1 out of 16 dinucleotides is a TT, right? Solar energy, ultraviolet energy, can convert those T's into a cross-link. They're supposed to be base pairing with A's, but they can get a covalent bond between the 2 T's. And that's bad. That has to be repaired. It turns out that you need NAD to repair that DNA damage. And when your NAD is being committed to that DNA repair process, can't do all the other things that NAD is supposed to do.
Right. Okay.
We showed in 2020 during the onset of the COVID-19 pandemic that coronaviruses through the double-stranded RNA genome, which is recognized as foreign, activates 5 different enzymes that churn NAD. They're called members of the PARP family. And 5 different members of the PARP family get excited and start churning NAD when in the presence of the double-stranded RNA. So that's part of the innate immune response. So when our innate immune system gets activated, we basically churn NAD. And then in addition, and it's not really known whether it's just intrinsic aging like a timer or whether it's all of the episodes of metabolic stress that we experience in our lifetime. But there's ample evidence now that NAD, declines in aging in a number of tissues. And so that's basically the use case for supplementation with NAD precursors.
Well, evidently, yeah. Because if it is declining and it's an essential process for many of these aging diseases, then why wouldn't we want to preserve NAD?
So the first indication for supplementing with NAD precursors was basically 100 years ago.
Such as NMN and NR, the precursors.
NMN becomes a precursor. NMN is used as a precursor. It's not actually a vitamin because it has a phosphate on it. But the 3 precursors are actually NR, nicotinamide, and nicotinic acid. Tryptophan being the de novo precursor. And then the 3 vitamins are NR, nicotinamide, and nicotinic acid. NMN absolutely is being used globally as an NAD precursor. It's technically, like I said, it's more like somebody taking coenzyme A to elevate their coenzyme A. It has to be degraded to something. Maybe that's too inside baseball for this discussion. But what I was going to tell you is that 100 years ago in the American South, there was a very prevalent nutritional disorder called pellagra that affected like 1 million people. And you don't really see it anymore. You'd have to be a doctor working with people in very small towns in India or various serious alcoholics to ever see a case of pellagra. But in the American South, there were basically poor farmers that grew corn and were provided with metal corn milling machines to make the ground corn maize, right, that they sold back to the people selling them seeds, right? It's kind of it's a very challenging economic model, right, where they have to buy seeds and buy equipment and then sell the stuff back. And basically their diet consisted of corn rations, lard, and molasses. Okay. And they had this presentation with diarrhea, dermatitis, and dementia.
Interesting.
And people in Washington, the way the American system works, these are small states. It's not New York, it's not California, but everybody's got 2 senators, Mississippi, Alabama, Georgia, where this was happening. And it went to Washington. They established the Public Health Service because of pellagra.
Okay.
This is the precursor to the United States NIH. They awarded the first Public Health Service grant to study pellagra. That's how important NAD is, it turns out. It was awarded to Joseph Goldberger. Everybody told Joseph Goldberger, you're going to go down to the American South and you're going to find an infectious disease that is affecting these folks. He goes down to these places. He goes to hospitals and prisons where there's a lot of people with pellagra. And in the prisons, he sees the prisoners have pellagra and the jailers don't. In the hospitals, he sees the patients have pellagra, the doctors and the nurses don't. And he said, this is not an infectious disease. And he looks at, the doctors and nurses. I take my coffee black, but a lot of people put milk in their coffee, and the doctors and nurses put milk in their coffee, and they have fresh food. They have eggs, they have meat. And the people with pellagra have corn rations, lard, and molasses, which, by the way you grind corn in Mesoamerica, because corn was indigenous to the Western Hemisphere, you grind corn with stone, okay, with alkali stone. And that extraction process will liberate NAD precursors. But if you grind corn meal with these metal corn milling devices that they were sold, there's someone on Twitter named Sarah Traber that kind of teaches this whole lesson. If you grind corn with this milling device and then you don't have any other fresh food, you're going to be deficient in NAD precursors. So Goldberger actually died without figuring out it was NAD precursors, but he figured out it was a nutritional deficiency and it could be cured with fresh food. And basically all fresh food has NAD precursors because, like I said at the very, very beginning, NAD is the electron carrier in all of life. So whether you're eating fruits, vegetables, fruits don't have as much cellular stuff as as beef liver, but virtually all fresh foods have some NAD precursors in them. And so you're eating fresh foods, you're getting NAD. NAD breaks down to the NAD precursor vitamins, and then you don't get sick.
And then we have energy. It's kind of, what I think about now is genetics versus epigenetics. Because when we look at, with 25,000 genes, if we look at it from a neurodegenerative standpoint, we know that you can switch on these ApoE4 genes, or you can just not switch them on, and you may get Alzheimer's disease, you may not, and it's all dependent on environmental factors.
Yeah, environment is huge.
Yeah.
Right. We have a gene set that can allow us to live to 120. Although we don't know quite how special the people are that are centenarians, but most of us probably don't live to our genetic potential because of environment.
Well, that's what I want to get into now. That was a really brief overview of what I believe that you really do know when it comes to NAD. I think if we did have 8 hours, we could sit here and talk more on that. But now I want to kind of move the discussion into longevity. And I want to first open up by asking, why do we age, first and foremost, and why can't we live to 200 years old?
Why we age is a tough question, but we do have genetic instructions, right? And all living things have genetic instructions. A bacterium is not going to be a raccoon, and a raccoon, is not going to be a bacterium. Although raccoons will harbor bacteria in their gut, just as we do. And but our genes, they specify the sequence of all of our structural proteins and enzymes, and there are what are called morphogenetic, regulators. So animals have something called Wnt genes that give us a front and a back, and a head and a tail, and a right and a left. And 5 fingers on each hand, and stuff like that. And then based upon the way particular species evolved, they evolved with a particular life trajectory, let's call it. Okay. And it can differ widely within closely related species. So a good example is, you're coming from New York City, so you might have a New York City subway rat in mind. If I say the word rat, which is big, but there's something called a naked mole rat that is basically the size of a house mouse, and we use these mice extensively in molecular biology.
That's pretty much because they're the closest thing to the human.
Not really.
Not really?
I mean, the closest thing to human would be a chimpanzee.
Okay.
And but it's a size where you can put 5 of them in a cage, and there's tremendous genetic tools to do mouse genetics. Basically, is the reason. But we can actually argue about the strengths and the weaknesses of mouse models of various diseases and conditions. But a mouse and a naked mole rat are basically the same size. They have the same basic body plan. They have the same rough number of genes. And naked mole rat lives 10 times as long as a mouse. 10 times as long.
Why?
And it is adapted to a environment in which the species evolved to be able to mate at 25 years old.
Right.
And basically all of its genes, it's not one gene, It's not one magical gene that converted a mouse to a naked mole rat and gave it this 10 times longer lifespan. It's thousands of different genes that give a degree of resilience to the respiratory system, the circulatory system, the skeletal muscle system, the brain, the hearing system, the odorant detection system, all of the systems of the naked mole rat that allow it to live and reproduce and take care of itself for 25, 30 years. And by the way, humans are kind of more similar to the naked mole rat than the mouse in the sense that humans are extraordinary good agers. So there's a book I reviewed. I reviewed a couple of books on longevity, and the one that I really liked was by Stephen Austad. It's called Methuselah's Zoo. And the idea of Austad's book is let's study animals that are really good agers. And earlier in his career, he drew a line, okay, where it's the animal's weight in kilograms along one axis and their lifespan in the other axis, I think the y-axis. And generally, most animals, the longevity kind of scales with size, right? So you have huge whales that have very, very long lifespans, right? Yeah. And you have little, mice and beetles and flies that have really short lifespans. And you put hundreds of different species on this thing, it approximates a line, right? And then what Austad and his trainees realized is if an animal is above the line, i.e., it has a longer lifespan than you would expect from its size, it must have figured something out about aging, right? So the naked mole rat is above that line. Humans are above that line.
Right.
Okay. So our closest, relatives are chimpanzees and like great apes, right? Gorillas. So gorillas are bigger than us. Chimpanzees are a bit smaller than us, but we live longer than both, right? Gorillas don't outlive us, even though they're bigger and heavier, and we live, quite a bit longer than chimps. And if you look throughout the animal kingdom, generally speaking, with some very interesting exceptions, generally speaking, animals live as long as they're reproductively capable, because basically that's what is selected in evolution, right? Is that, someone said something ridiculous the other day, on Twitter. Someone said something ridiculous on Twitter and said that humans were only designed, which is a weird word to use, to live 30 years. And, 100,000 years ago, the grandparents would be like stealing food from their grandchildren. No, no. If there was a species that stole food from its babies or grandchildren, that species would not exist.
Yeah, there's a grandma is it grandma theory?
Yeah, let me get there in just 1 second. But that is literally the exact opposite.
Was this person in science?
You can look it up.
Okay.
But strangely enough, the person gives very expensive seminars on longevity. People pay money, for the person's content, which is weird. But the species did not succeed by stealing from our babies. The species succeeded by either fecundity, which means having lots of babies, and or caretaking, meaning we have lesser number of babies, humans being the extreme example, right? Where there's a tremendous amount of caretaking of the offspring. And then, bam, you hit the nail on the head that the most interesting exception to the rule of an animal that outlives her reproductive capability is the human female.
Is the human female.
The human female, right? Average onset of menopause, 51. Most babies born by the time she's 35 or 40, right? Much higher fertility in her 20s than even in her 30s and 40s. No judgments here, but I can show you the data.
But can still conceive
But
in there.
can conceive. But absolutely can conceive. I mean, onset of menopause is not till 51. But life expectancy into the 80s. So 3 decades plus, right, of good health, better life expectancy than men at almost every decade of our maturation. Better health in her 50s, 60s, and 70s and 80s than men. Yet, onset of menopause is at 51. So I just told you that in general, the selection in animal, lifespan, the selection is, there's a lot of selective pressure for us to be born. I mean, every animal, okay, beetles, flies, birds, lizards, we have to be born, we have to avoid predation, right? And then we have to be able to get to the point where we can get our own food. Then we have to get to sexual maturity, and we have to be able to identify mates. And so that requires a sense of smell, of mobility, vision, all kinds of things, right? Animal sex appeal, whatever that is. And then be able to reproduce. And then you either reproduce with lots and lots of babies or good caretaking of the babies so that your gene set, is passed on. And that's why we have the animals that we have.
Yeah.
Right? So we don't have animals that stole from their babies or their grandkids. That's literally not the way evolution works. We have animals that have lots of babies and or provide for their babies and grandchildren. And the grandmother effect, right, is that we think that in human evolution, because humans do live longer than chimpanzees, right, is that, but the, if there were a system that preserved, primary follicles, right, and fertilizable eggs that you could have into your 50s and 60s and 70s, it appears to me like it would have resulted in many more birth defects, right? And many more kind of less viable offspring, right? So in the course of human evolution, the reproductive period for females was kind of condensed from 15 to something in the 40s, right? 40s to 50. But then women are still very capable because they have played such an important role in child rearing and even provide advantages to grandchildren. So that's the grandmother hypothesis, right? That women's reproductive period was condensed into a period that is substantially before the end of her, lifespan. But in men, fertility just declines slowly. I used the term peter out once, and people cracked up. But it's true. Basically, there's a sperm count, that just declines, and there's probably a sex appeal that just declines. So there's not very many men that father kids when they're 70 or something, but it's possible.
Oh, it's very possible. I think can't men conceive to their 90s?
Yeah, if they live to their 90s, right? And that's basically the way most animals are. So
Yeah.
the Greenland shark that lives to 180 or 190 is reproducing when it's 180. It's an apex predator, right? There's not anything that it has to really worry about other than like boats or people. And it's got sex appeal when it's 180, which is why every aspect of its body works really well when it's 180. It's not one longevity gene. It's the difference between, a Greenland shark and other types of sharks that live much shorter is hundreds or thousands of genes that allow all of its systems to work really, really well for all of that time, because reproduction involves everything, right? Reproduction involves musculature, brains, being able to smell, vision, everything, right? So I think that's the way, evolution works. And that's why animals have the longevity that they have.
So then, okay, so why don't we talk about Surtuins then, since we're on this topic of longevity? I know that I've actually had David Sinclair on the podcast. This was a couple of years ago, actually, during the pandemic, but we spoke about Surtuins, and I have understood his theory of aging and longevity, and I'd love to know what your
Yeah, well, there's a group of people that have falsely claimed that Sirtuin genes are conserved longevity genes. They're not. And
Actually, I do want to read your the title of your wonderful paper that was released, I think 2022, October or September 2020. Surtuins are not conserved longevity genes. I had to tear apart this study, highlight it about 6 or 7 times. So you're basically saying that Surtuins are not longevity genes.
No. So the original result was from the 1990s in the lab where David Sinclair was a postdoc, Lenny Guarenti's lab. There were a number of, very excellent people that trained there, and they were using yeast. Remember, I told you that I did my graduate work on a yeast enzyme. I got nothing against yeast. I did yeast when I was in industry. I did yeast for the large part of my initial independent academic career. Yeast has taught us important things about the cell cycle. It's taught us important things about cancer, believe it or not. It taught us things about prohormone processing. And the Guarenti lab thought that it might tell us something about aging. I think yeast aging only tells us about yeast aging, unfortunately.
Yeah.
And there are 2 different models of yeast aging. There's one model where you look to see how long cells live, right? So we can go back to my example of making pizza. So the yeast in the jar are dried yeast, right? I don't know the last time that they were dividing, right? I don't know when they were put in the jar. I bet they were put in the jar a year or 2 ago, right? And I gave it water and flour and a little bit of olive oil, and that's fuel, and it got bubbling right away, right? And so if yeast had a short lifespan, that's called a chronological lifespan, we wouldn't have yeast.
Yeah.
Right? We've literally been carrying around yeast in our, clay pots for thousands of years.
Okay.
Right?
Yeah.
So if you look at agriculture, development of bread making, wine making, beer making, we're going back to Mesopotamia, and other places in the world, and that's how long we've been cultivating yeast. So that's aging, right? So you can see how long yeast will survive when you basically dry it down and you let it sit for a while. And then there's another assay of aging that was done by the Guarenti lab, and it involves taking individual cells and putting them on a petri dish. Okay, so no one would you wouldn't ever do this for making pizza, right? But you could put individual cells on a petri dish, and then you can kind of like install a graduate student or a postdoc in front of the petri dish and microscope. And then every 90 minutes, the yeast cells divide. And what's called a mother cell will produce a daughter cell. The yeast divide by what's called budding. It buds into a daughter cell. And if you remove that daughter cell, you can see how many times they're capable of producing a daughter cell. That one cell that you arrayed, you might have 50 individual cells on a petri dish, and you can determine how many times the mother cell produces a daughter. And the answer is around 21 times. And if you calorie restrict it, if you have lower glucose, lower sugar on the plate, it will go longer. It will, divide more than 21 times. But if you go back to the yeast culture, you'll never find a yeast cell that is divided 20 or 21 times. That's literally 1 out of 5 million cells is that old in the culture. 50% of the cells in a culture are brand new cells, right? They just budded off. Mm-hmm. So 50% of the cells have never been what's called a mother cell. 25% of them have been a mother cell one time. 12.5% have been a mother 2 times. It's an exponential series, right? And so the effect of Sirtuin, the original Sirtuin gene was called yeast Sirtuin. The effect of this Sirtuin gene was to allow, in this one particular assay of aging, it was to allow those 50 arrayed mother cells to divide not 21 times, but 25 times. But this is an effect of an incredibly rare cell. 1 out of 5 million cells is providing an advantage of 1 out of 5 million cells. Okay. And I want to go back to the jar of yeast, right? If you do the assay and that represents basically the whole culture, right? You grow yeast and you look at their ability to be revived. It turns out if you delete the Sirtuin gene, that culture lives longer chronologically. It's literally the opposite. Yeah. It's literally the opposite of an anti-aging gene in one yeast aging assay. Yeah. And then it provides an advantage for 1 out of 5 million cells in the assay that they used.
So then what was David trying to achieve with this Sirtuin gene?
Well, basically they had this idea that because they found advantage. Actually, Matt Kaberlein, I did some of the.
Him on the podcast.
Matt did some of the very, 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 very first experiments with Sirtuin in yeast, and then David in 1997 showed that it had to do with what's called ribosomal DNA circles, right? And ribosomal DNA circle, we don't have ribosomal DNA circles in humans, and worms don't have them, and flies don't have them, and so they had a very peculiar idea that because they found, it would have been great if it were true, that the gene that they found in their particular assay of yeast aging, they proposed that this is going to be an anti-aging gene in worms and flies and mice and people. And here's the tragic part. There were irreproducible positive results published in the
Yes.
early 2000s.
Mm-hmm.
So the Guarenti lab published that extra copies of the worm Sirtu homologous gene extend lifespan in worms. And then David Sinclair, in collaboration with a fly group, claimed that extra copies of the fly Drosophila Sirtu gene extended lifespan in flies, which to me that would be mind-blowing, right? Because yeast is a fungi. Yeast evolved its whole lifestyle before there was any animal on this planet. So the idea from this Guarenti, Sinclair set of experiments is that somehow, whoops, I just almost caused a little accident here. Got so excited. But the mind-blowing, concept is that fungi have a gene, like yeast has a gene that anticipates the causes of death and the causes of aging in animals, and that you have extra copies of that same one gene in animals that's going to extend lifespan. And then they got results that seem to be in support of that.
But couldn't produce the results.
They continue to claim that those results are true. Other people cannot reproduce those results. That's a bad sign.
Yes.
If you're finding something that is authentically true, other people have got to be able to reproduce it. It's like this verdachte thing about the semiconductor about 2-3 weeks ago, right? People are trying to reproduce it because if it's really true that there's room temperature superconducting, it's going to enable all kinds of, high-speed trains and all kinds of things that we don't have right now, right? But no, it turned out to be conventional, magnetism. And so it's really a problem.
Yeah.
And, and further, David, claimed to discover resveratrol as a Sirtuin activator, and it's not. Resveratrol, it's what's called a biochemical artifact. So there's a synthetic peptide that they used to do that assay. Resveratrol interacts with the synthetic fluorescent piece of that peptide. It does not interact with Sirtuin. The company that they established.
Has he retracted that now?
No.
Though? Oh, okay. He hasn't.
No,
okay,
he's still, today in 2023, he's got a business that sells resveratrol to the general public.
And that's ludicrous.
And, and claims that it's activating Sirtuin and that Sirtuin is a longevity gene. And there's two false statements there. It doesn't activate Sirtuin, and Sirtuin is not a longevity gene. So it's astonishing to me.
How is that? Okay. That's interesting that it's such a big public name is out there doing that if you can conclusively say that Sirtuin is not a longevity gene.
Sirtuin is not a longevity gene in worms. It's not in flies. It's not in mice. And it's not in people.
Are any of the Surtuins?
Sirt6, all of there are 7 Sirtuin genes. They're all, interesting.
What does it stand for?
It stands for silent information regulator. So the original yeast gene was found by somebody named Jasper Rhine. He's a yeast geneticist, Rhine and Herskowitz. It's from, I think it's literally from Jasper Rhine's 1975 PhD thesis, where he's looking at yeast mating type. Believe it or not, there's 2 mating types in yeast. It's not male and female. It's something called A and alpha. And there's a way that yeast don't express both mating types. There's a way that yeast are not bisexual, basically. And it involves the Sirtu gene. Okay. Repressing the opposite mating type information. And then the kind of Sirtu thing was kind of like rediscovered with the so-called anti-aging phenotypes in the 90s in the Grenti lab by Matt Kaberlein, David Sinclair, and others. But the problem is, like I said, is that it's only one of the two aging assays in yeast. It only advantages 1 out of 5 million cells. It has the opposite effect in chronological aging in yeast, which was done by Walter Longo, who's a very prestigious aging researcher here in Los Angeles. And then it doesn't translate into animals.
So that Cert6 gene, is there an activator for that?
So Cert6, in some people looked at Cert6 because they thought that Sirtuins were longevity genes. So it's sort of like looking for your keys under the street lamp, right? If you lost your keys, somewhere in a 1, mile radius, right? And there happens to be one street lamp in the 1 mile radius. And, you might only look under the street lamp because that's a place where your keys would be illuminated if they were there. But the problem with looking at Cert6 or Cert2 or Cert3 or 5 or something is that there was a presumption that these are going to be longevity genes, right? That's a falsely premised assumption. But when people looked at Cert6, they found that in certain mouse strain backgrounds, you get a bit longer lifespan. To me, it looks like it's not going to translate into humans because Cert6 is a positive regulator of gluconeogenesis, right? This is this overnight process where our liver elevates our blood sugar, right? The problem is humans, as we age, we tend to have a worse metabolism and our blood sugar rises, which is why so many people take things like metformin, right, to lower their blood sugar. Again, I don't think that metformin is necessarily going to be a successful longevity drug, but there are trials of metformin as a longevity drug. Cert6 overexpression elevates blood glucose. That's good for a mouse.
Bad for a human.
Bad for a human. Mouse has too low blood glucose as it gets to 2 years old. A lot of humans have too high blood glucose as we get to 70 or 80 years old. So Cert6, is an interesting mouse, but I don't know that an activator of Cert6 is going to be that valuable to us.
Okay, what I would love to understand is how can we, in your words and everything that you've learned, how can we slow the aging process?
I think that we definitely can age better, right? You can age better and you can age worse. I think largely it's following mom's advice, to be honest, right? It's a good diet. It's high physical and mental activity, social engagement, sleep, don't smoke, don't do drugs, don't drink alcohol in excess, take medications that you need to take, don't take drugs that you don't need to take.
Drink clean water.
Clean water, right? And so that's almost all of it, right? I think that there's a use case for NR, excuse me, as a molecule that promotes resiliency and repair. I think that, in coming years, I think that currently we have human evidence that NR is anti-inflammatory and in combination with other molecules that it accelerates time to recovery of COVID. There's a lot of long COVID studies that are being done. There's potential for wound healing and fatty liver studies in which we may get some positive results. There's a lot of anecdotal data that people, have better workout recovery and recovery from minor cuts and scrapes, and that potentially you age better and you can kind of maintain your muscle mass with good exercise and NR. But I don't necessarily believe in magic pills. You know, if there were single longevity genes, dominantly acting longevity genes, then CRISPR would change a lot, right? Because if there were a dominantly acting longevity gene, then we would want to like modify our genome to have more of it, right? Or we would want an activator of that gene. That was kind of the resveratrol idea, right? But, you know, Sirtuin isn't a dominant active longevity gene. And the most powerful single genes that affect longevity in animals are actually recessive alleles of growth promoting pathways. You know about that? The dwarf mice business, right?
Yeah.
So like there are dwarf mice, they have basically mutations in pituitary pathways. And then these are tiny little cold mice that can't compete for food and are infertile that live twice as long as other mice. So, and, you know, no one wants to, I don't think there's a lot of humans that would want the difficulty of being a dwarf mouse, right? Because those are not happy little campers. And in fact, if you go to the things that are called longevity clinics, so here we are in West Hollywood, you could go into, you know, clinics in order to look really good for your next movie shoot or something like that.
And they would hook you up to an IV, an NAD IV.
Well, they would potentially give you growth hormone and anabolic steroids. And basically these mice don't have growth hormone and they live longer. And we know that pro athletes that have taken growth hormone and anabolic steroids become relatively fragile, you know, much more rapidly. And so it's probably, you know, it's not clear yet that you can directly target the aging system, which is kind of a goal of geroscience. It would be wonderful if we could. We can target the NAD system and, you know, potentially enhance people's resiliency and repair, but I don't think that we're necessarily going to extend lifespan that way. I think that our gene set is, you know, limiting our lifespan. We potentially are going to enhance our health span and a resiliency and a repair, but that's something that has to be demonstrated in, you know, placebo-controlled trials.
Okay, so mainly what I want to know now is there is just so much happening on social media, which I'm sure this is the reason why you're probably not on Instagram. And for your sake, it might hurt you if you go on there and see the abundance of information out there, albeit most likely not true, around longevity. This helps you live longer. This helps you live longer, even back to the resveratrol saga. You know, I do believe David was out there also suggesting a glass of wine to increase resveratrol levels to help with longevity, which I think is also scary. What currently is false? What are we reading that is false around longevity?
You know, none of the thing is, I don't even know where to start.
Okay, give me the top two.
There's a lot of fads, right? There's a lot of fads in diet, you know,
Like fasting.
like intermittent fasting, right? Yeah. And then there's extreme practices of all sorts, and there's people that are highly prescriptive about all sorts of, you know, physical activities. So basically, first of all, you have to live your life, right? And you have to figure out what works for you. There are people that are, claiming that we should all be doing intermittent fasting or some type of calorie restriction. So, calorie restriction is a way to lose weight and, effectively. And so you have to be in a caloric deficit in order to lose weight. One of the ways that your body is going to respond to that is by lowering your energy expenditure. Okay. I'm personally at pretty much the weight that I want to be. And so, caloric restriction doesn't really make any sense to me. In terms of intermittent fasting, I'm on team breakfast. And, I am very productive when I get up in the morning, have my coffee and, have breakfast, and I can get to work and I can have a very good workout and I can have good mental, capacity. And so there are people that are just highly, highly prescriptive about the way you need to live your life that I don't find particularly helpful. On the other hand, overweight is a huge problem, right? And, I work in a cancer center, and if you look at the global statistics, there's essentially only one organ site that is becoming more prevalent in cancer and more lethal in cancer, and that's liver cancer. And that's because the lesion that predates most hepatocellular cancer is fatty liver. And there's something like 1 billion people on the planet right now that have fatty liver. And the percent of the people with fatty liver now that become type 2 diabetic or, develop liver, cancer could, further degrade human life expectancy and, really challenge global economies because it takes, so much, medical care to take care of people with type 2 diabetes and hepatocellular cancer, and it's incredibly lethal. HCC is incredibly lethal. So for the first time ever, there's actually drugs that actually make calorie restriction possible, sustainable for most people, right? So these GLP-1 receptor agonists like semaglutide and so forth. So that's exciting, right? But that's indicated for someone that is living with overweight that that wants to be able to restrict their appetite. So
so calorie restriction.
Caloric restriction makes sense, but caloric restriction extends lifespan of caged mice. It extends lifespan and health span of primates as well, but a lot of the comparator groups are actually overfed, right? So caloric restriction, the question is always compared to what, right? So, I think that most people, would probably do better by eating more plant matter and drinking less sugar sweetened beverages. But I'm not going to go around and advocate caloric restriction on the internet because some of the people that are most likely to take that advice are already really lean and they're basically the worried well, right? And somebody that is living with overweight pretty much knows that they need to move more and eat less, but may not be able to do that, right? Okay. So I don't see the point in being an advocate for caloric restriction. There's a lot of people that are sensitized to advice on the internet. I call it kind of podcast disease. Yeah. The present company excluded, but there are people that listen to podcasts to try to get every little bit of advice that they could use to so-called optimize their life. Somebody that tells them when they should have their coffee in the morning. I don't understand why we need to give that kind of advice.
Why we need to wait 90 minutes after waking up to have our coffee?
We 5 billion people or 7 or 8 billion people on the planet have different needs in the morning. Some people have childcare. Some people, have to get to work in 30 minutes and don't have 90 minutes to go and take a walk and wait until the sun is at 15 degrees above the horizon. Some people live in the, near the North and South Pole and near the equator where that 15 degrees above, the horizon is, a different time of day. And, you can make advice for people, but it doesn't make sense for vast numbers of people to follow it. And I just think there's too many people that are too prescriptive about stuff, and that people have to live their life and come up with a sustainable practice, like exercise, for example. So, the people that I like to follow that talk about exercise and fitness are all about sustainability of the activity.
Of course, because adherence, we know that adherence is the best form of consistency, which equals,
so there's some people that are saying, okay, well, should I do like 3 reps to exhaustion, or should I do multiple sets of 10 or something like that? Well, if you're at that level of refinement, you're good.
Just pick up the dumbbell.
You're good.
Yeah.
And then, because it turns out that walking is great, running, swimming, mountain climbing, bicycling. I don't have a standing desk because it wouldn't really work for me. But treadmill desk, that kind of thing. A lot of those things are gimmicks. If it works for someone, that's great. But chasing around kids, that counts. Walking your dog, that counts. Being around people of a variety of ages, taking care of other people, that's a physical activity. So, there are people that will point out that if it feels like, play, and if it's meaningful to you, and you do it, then that's the right activity. So, I think that people have to find their thing that they can stick with and then do it. But, support groups, coaching, whatever it takes.
I want to finish up by, I think I've asked you this question 6 or 7 times already in the episode, but I do want to get to the bottom of these IVs and how to actually increase levels of NAD. I mentioned offline that there are companies who now are selling NAD powder as a supplement.
Well, it's the same problem as NMN, right? So it basically has to degrade to NR,
okay.
right? And safety is really paramount here, right? So you talked about your food and water being not contaminated, right? And so if you buy supplements, you don't necessarily know what's in them unless they've gone through a rigorous program of testing. So we did that with NR. So I discovered the vitamin pathway, and then a number of years later, it was, the IP was licensed and commercialized, and we did the human testing, right?
Is that true, Niagen?
Yeah. So Niagen is like the branded, patented NR, and we know that's safe for humans, and it's going to deliver the same thing that NAD or NMN do, which is going to deliver NR to cells. Now, one thing that I didn't say is why. So I said that tryptophan to NAD is an inefficient process because tryptophan is used for other things. It requires a lot of ATP, and it doesn't work in all tissues, right? And neither does nicotinic acid, niacin. Niacin also gives you a high flush reaction if you take high dose niacin. The nicotinamide pathway and the NR pathway are active in all tissues, But the interesting thing about NR is that in many of these conditions of metabolic stress that I discussed, like the coronavirus infection and in neurodegeneration and in heart failure, and in, we think in aging as well, the NR pathway is upregulated. So it's like your cells and tissues are looking for NR when the NAD is being depleted. So we think that that's the added value of NR, that it costs less ATP for cells to make NAD coenzymes from the NR pathway because it already has the ribose on it. And so that's kind of the use case of NR. So we think that you can age better with NR, and it will, promote resiliency. In terms of IV, I don't really know where they're getting the IV. I don't know how clean that whole, system is, but all it's going to do is going to deliver NR. I've heard it's relatively painful as well.
Very painful.
And so I think what it is an inflammatory reaction because normally NAD is an intracellular metabolite. So cells have an innate immune response, just like they have an innate immune response to double-stranded RNA because it's interpreted as a viral product. Cells get kind of excited when they see things like extracellular ATP, NAD, intact mitochondria, cell-free DNA. And so that produces an inflammatory reaction. So I think it may be painful because it's producing this inflammatory reaction. So I don't really see a use case for intravenous NAD. You can basically get the effect from oral NR.
Wow. No matter how many podcasts I do, which I think I'm around 300, the answers really are sleep, eat well, social connection, sunlight, and exercise. It's, at the end of the day, I feel like we're spending billions of hours, billions of money to try and find the answer to this evolutionary process that is just, it's just so nuanced and just, I feel like the answers are just eat, sleep.
What mom
what
said,
my grandparents.
Right. Exactly, yeah. Right. I don't really see a tremendous amount of human longevity tech right now. Like I don't see the consumer use case for age tests. They don't agree with each other. They don't measure what they report to be measuring. They're sensitive to inflammation. So they'll say that you gained 10 years during a COVID infection and then you lost 10 years when you resolved the infection. Okay, so let's admit you're measuring infection. You're measuring inflammation. Okay. And so and then there are people that are trying to turn every single biomeasurement, every single biomarker into an age. There's a person out there that says his testosterone age is 18, even though he's in his 40s. But guess what? Yeah, you're juicing with testosterone. So you're not 18, you're juicing with testosterone. So they're making a mockery of these biomarkers because the whole thing can be gamed, right? You sell somebody a kit to measure their age, and then you give them these various supplements and drugs that are basically goosing the age test. So I'm basically a very, very healthy 61, okay? But I'm 61.
Wow. Yeah.
And I'm going to be 62 in a couple months. And, but I chase around a couple kids. I have a wife that is a marathoner, and, I have a lab full of young people, and, I have people that depend on me. So I'm active, and I'm, but I don't see a use case for taking an age test that gives me some number for me to brag about. It's function, right? It's, can you still do a pull-up? Yeah. Can you, do you have a sex life? Can you play chess with somebody, half decent? Can you ski? That's what matters. And to me, the age tests is kind of a racket. It's also a research tool. Okay, the age tests are a research tool, but there's not a consumer use case for them.
I think Brian Johnson would disagree.
Well, he's literally selling, a plan.
Yeah.
But it makes no sense. I mean, the things that he's doing that are making him more fit in his 40s than he was in
saying
his 30s.
Saying that he's got a biological age of 18, I think it is.
Well, no, but it's tests that are totally biased by the supplements and drugs that he's taking.
Yeah.
So like he literally tells you his testosterone age, and then if you look in his protocol, it's testosterone.
Yeah.
So it's not meaningful. The things that he's doing of eating lots of veggies and working out and prioritizing sleep that's his functional fitness, right? And then then you can add hair dye and laser treatments to things, which is superficial, and other things that are just not going to work out for him that well in the long run, because testosterone replacement therapy will, if you're a guy, literally shrink your balls and lower your body's endogenous production of androgens and potentially accelerate aging. So, he's measuring himself to try to have a higher level of testosterone and a higher level of growth hormone. And I told you the one thing that we learned from one of the most powerful things that we learned from mouse, aging studies is that the growth hormone deficient mice live longer. So if people that are juicing with growth hormone and testosterone are going to, feel they're going to be able to put on muscle mass, maybe like they did in their 20s and 30s, but are probably going to be aging more rapidly. So it's not evidence-based what he's doing.
He may be coming on the podcast, so that's going to be interesting. Okay. Charles, this episode is probably one of our longest and most in-depth. I think people need a pen and paper for this one. Are you only on Twitter? I want to point people to your work.
I'm on Twitter. I mean, I'm on Instagram and Threads and most of that other stuff, but I'm not. That. Yeah, I'm not really active on it. Okay. So the only place that I've really got a substantive following, I think, is on Twitter. Yeah.
We're going to point everyone to there. And okay. I'm very excited to follow your work. I think that we'll have another podcast next year just to catch up on all of this.
Good. People can really, if they want to really follow my work, there's a website. It's brennerlab.net. Mm-hmm. And, that's where our publications are. And but I try to let people know if I'm giving a talk that's open to the public. I try to let people know on Twitter, and people can ask me questions there.
Thank you so much, Charles.
All right, thanks. It was a pleasure.